Hello everyone. Welcome to special IBM webinar for Indian researchers. I'm Virgon from IBM Technology and I'm glad you could join us today. In today's session, we'll be discussing real-time intravital confocal and two-photon microscopy (IVM) for in vivo cellular level imaging of internal organs in live animals. We'll explore how advanced two-photon and confocal microscopy enable high-resolution in vivo imaging and reveal cellular dynamics across organs with application in drug delivery and disease research. Now, I'd like to introduce today's presenter. Due to an unexpected health issue, Hayden won't be able to join us today. We're so sorry about that. Instead, Jenny from our technical application team will be presenting. Jenny leads the animal experiment operation for our CRO imaging service within the sales division. If you have any question about surgical procedures or animal preparation, she's the ideal person to provide in-depth insights. As you follow Jenny's presentation, we invite you to explore our website, ibmtech.com, and shop page, ibmtech.shop, to discover more about the imaging solution featured today. And please don't hesitate to submit your question in the Q&A box at any time during the presentation. We'll do our best to answer as many as possible during the Q&A session at the end. And Jenny, if you're ready, please go ahead with your presentation.
Hello, everybody. Thank you, Morgan, for the introduction. I'm Jenny Ohm, Application Specialist from IBM Technology. Thank you very much again for joining today's webinar and for your great interest in our intravital microscopy technology. I'll begin with a brief introduction to intravital imaging, followed by an overview of our systems and several application examples. You may find that many of these approaches can be applied to your own research, so please feel free to leave any questions in the Q&A box and we'll address them one by one after the presentation. So let's start with a very brief overview of intravital microscopy. So intravital microscopy, or IVM, is a powerful imaging technique that lets us watch how cells and molecules behave in real time inside living animals. Unlike traditional methods like histology or in vitro and ex vivo methods, IVM shows what's happening in a very natural living environment, so we can actually see things such as how cells move, interact, and respond during disease progression. Compared to conventional imaging systems like the X-ray or IVS, IVM offers something very different. It gives us high-resolution, real-time images at the cellular level. This makes IVM a very valuable tool for today's pharmaceutical and biotech research, where dynamic and detailed insight is more important than ever. So in vivo imaging modalities allow us to observe biological processes in their natural living environment, something traditional methods like immunohistochemistry cannot achieve. With these techniques, we can directly visualize how cells move, interact, and respond during any disease progression, as you can see here on the right. This is an example of the lung imaging done with intravital microscopy, and we can see at the cellular level how the immune cells were interacting within the lung vasculature after being induced by sepsis. And as mentioned previously, traditional in vitro and ex vivo methods only provide very rough and low resolution data as they capture images at the whole body or the whole organ level. So to overcome these limitations, IVM technology has designed a much more advanced intravital microscopy system that allows researchers to track cellular dynamics, observe drug distribution and therapeutic effects, even in very challenging organs such as the lungs or the heart in all living animals. A very distinct strength of IVM's intravital microscopy system lies in its departure from conventional in vivo imaging approaches. So as mentioned, traditional methods require the preparation of separate animals for each time point, inherently introducing substantial inter-animal variability. In contrast, our microscopy system enables longitudinal imaging within a single living subject at precisely defined time points, dramatically enhancing consistency and reliability. More than just static image acquisition, it empowers real-time dynamic visualization of biological processes as they unfold naturally in the living body. So to better understand how our body works or the body of a living animal, such as mice, works, it's important to look at what's happening inside, as mentioned before, right down to the cellular level, especially in areas like immune response, cancer, and drug delivery. So intravital microscopy allows researchers to observe those changes in real time while they're happening. So IV technology system especially makes it possible to study how drugs travel through the body and how they affect cells in real time. For example, on the left, we can see the results from a sepsis model where, as mentioned before, inflammation had inhibited normal blood flow in the lungs. And on the right here, we tracked exosomes, anti-cancer drug movement, and transplanted cells in the animal body using intravital microscopy. So if we look more closely into IV Technologies' intravital microscopy system, it is provided as an all-in-one microscopy system designed for high-quality intravital imaging performance. This compact single-box package allows for easy installation, operation, and maintenance, allowing researchers to solely focus on imaging. And the system combines co-optimized hardware and software, featuring an ultra-fast polygon scanner, a temperature monitoring system, motion compensation technology, and a separate AI-based denoising software for stable and high-resolution imaging. Most notably with its automatic, hassle-free motion compensation technology, live animal maintenance tools, and various imaging window options, researchers can obtain high-quality in vivo data across a wide range of organs. Now let's take a closer look at some of the key features. First of all, I'd like to emphasize the dual usage of the confocal and two-photon laser of the intravital microscopy system. So now in the slide, you're seeing the imaging data of the neuromuscular junction from a Thigh1 YFP transgenic mouse, showing endogenous GFP from the nerve here. We additionally labeled the blood vessels with our in vivo labeling kits, which is shown in a blue color in this slide. Now, I'd like to share with you the same imaging data of the white dotted area here, but imaged with a two-photon laser. So without need of another in vivo labeling step, we could successfully observe the actin myosin structure by utilizing the second harmonic generation signal that naturally occurs when using a two-photon laser. And for this image here, we use a 920 nanometer wavelength two-photon laser. Notably, even using only the fixed 920 nanometer wavelength available with IVM's IVM CMS3 system, it allows for simultaneous observation of up to 4 different fluorescent signals, all in a single imaging acquisition. So if your research needs to see, for example, the accumulation or biological changes of fiber, collagen, or muscular structure, two-photon laser imaging would be the perfect choice for you. And another key distinction of the system is that it is equipped with an ultra-high-speed polygon mirror scanner, capable of achieving imaging speeds of up to 50 frames per second, leading to capture the dynamics of fast-moving cells like red blood cells or neutrophils flowing inside the blood vessels of the heart, as you can see in this example here. And especially this ultra-fast polygon mirror scanner maximizes the motion compensation technology algorithm, which enables high precision of motion artifact by facilitating frame-by-frame compensation. This is a highly optimized solution for imaging rapidly moving organs, such as the heart and the lungs, as it can compensate the X and Y movement artifacts with real-time acquisition of compensated data right away. And in addition to the motion compensation function, to maximize the utility of the video rate imaging capabilities of iBeam's microscope, we developed an AI software to efficiently denoise real-time videos obtained by intravital microscopy. So as a result, you can see that the software successfully, here on the left, preserved the true fluorescent signals and the high temporal resolution of the video while removing background noise. So on the right here, you can see the raw video of the processed data shown on the left, and you can see that there's a lot of noise, whereas on the left, the noise has been successfully denoised. And especially with various apparatus, which we call imaging windows and chambers, users can easily gain highly qualified longitudinal in vivo images from each internal organ. We are usually optimizing surgical methods for securing a cranial imaging window to the brain, allowing observation of various dynamics such as brain cells and drug permeability across the blood-brain barrier, and to observe various cellular or pharmaceutical dynamics within abdominal organs. You can acquire the imaging data from the abdominal imaging window implanted into animals for up to a month approximately. And if you are interested in observing any other organs that are not listed in the slide here for long-term studies, please feel free to reach out to us for potential collaboration, development, or optimization. And based on the advantages of being able to image almost every organ in a living animal's body at the cellular level, especially thanks to the windows and chambers mentioned before, iBeam Technology's intravital microscopy system can be used for imaging various disease models, including cancer models, along with the dynamics of immune cells. So not only can it observe processes such as proliferation and invasion of cancer, but it can also track immune cell responses and also observe the biodistribution of drugs as well. Now I'd like to move on to some of the actual application studies that we've performed at iBeam Technology using the intravital microscopy system. To begin, I'll show you how we visualized immune cell responses and the migration of immune cells in different organs using intravital microscopy. So first, we employed a lyse GFP transgenic mouse, which exhibits endogenous GFP expression, specifically in neutrophils and macrophages, to observe dynamic immune cell responses after inducing skin irritation by irradiation. Additionally, we utilized an in vivo labeling kit consisting of CD31 antibodies conjugated with far red fluorescent dyes to stain and visualize the blood vessels in the mouse ear skin. So as you can see here in the video on the right, we could successfully observe the blood vessels in the mouse ear skin. In the time-lapse video captured at 30-second intervals, we could also observe the active movement of immune cells around the ear skin vasculature as well. And here on the left, you can see how the mouse was prepared for the ear skin in vivo imaging. It is mounted onto the intravital microscope, and here you can see a holder has been used to secure and stabilize the ear skin during long-term imaging. So through this experiment, we identified dynamically moving immune cells in real time. And following this, we stimulated the skin of the same mouse with irradiation and observed changes in certain immune responses. So as shown in the image on the left, we found that the cell movement was significantly more active after irradiation compared to before. And we were also able to confirm this through further quantitative analysis, as you can see here on the right through the quantitative analysis graph that we created. So in the second skin imaging experiment, we used a CX3CR1 GFP transgenic mouse, which also expresses endogenous GFP, but in monocytes and in macrophages, to visualize immune responses, again, in the mouse ear skin. So a wound was induced with a 31 gauge needle puncture and a KRS inhibitor known for disrupting cell-to-cell adhesion, and it was applied 4 times to the area of the ear skin. So imaging immediately after the wound, as well as 1 and 2 days after, revealed a significant reduction in immune cell recruitment around the wound site in the KRS inhibitor treated test group compared to the control group here. So this reduction was attributed to decreased cell-to-cell adhesion and migration in the test group. And through quantitative analysis, we were also able to find out that only a third of the immune cells had clustered together compared to the control group here. So the next application study was done in the lung. So the lung tissue is especially difficult to image due to constant movement caused by respiration. So IBIM technology had developed a specific lung imaging chamber, which uses negative pressure to stabilize the lung in place, allowing researchers to capture high quality images in the lung. And not only that, but as mentioned before, using the motion compensation technology that is also embedded in the system, you can also achieve much more sharp and more accurate imaging images from your data, even when the organ, such as the lung, is moving very rapidly due to respiration. So here, we induce sepsis by injecting LPS to a lethal level. Then we labeled the blood vessels with FITC dextran and the neutrophils with Ly6G antibody-based labeling kits. And in the sepsis-induced group on the left, again, you can see that the neutrophils formed clusters in the lung vasculature, interrupting natural blood flow through the lungs. We were also able to analyze this quantitatively by measuring the velocity of blood circulation before and after inducing sepsis. Sorry. And so with IBIM's highly optimal imaging technique, you can gain much deeper insights into disease development, even in vastly moving organs such as the lung or heart, by utilizing the imaging chambers provided by IBIM technology, as well as using the Intravital Microscopy system. So now focusing more on long-term imaging, we visualize thrombosis induction and blood-brain barrier recovery in the brain. And through these studies, you may recognize the importance, again, of using imaging windows and chambers, which enable continuous, longitudinal, and real-time observation, providing valuable insights into disease progression and cellular responses. So in this particular study, we induced photothrombosis by injecting rose bengal intravenously into the mouse and irradiating the target area with a 561 nanometer wavelength laser. And using DHE and PI staining, we were able to visualize reactive oxygen species, or ROS, and cell death, confirming the successful induction of photothrombosis in the mouse brain. And this photothrombosis was conducted after we had implanted a cranial imaging window into the brain area of the mouse. So following photothrombosis induction, blood vessels were labeled and we imaged the area starting from day 0 to day 30. We initially observed signs of brain swelling, likely due to inflammatory responses that start at an early stage. However, by day 8, the imaging data revealed that the brain had contracted even more compared to day 0 here, as you can see on the left. So as you can see, the results highlighted with the yellow dotted box from day 2 to day 5, the swollen brain tissue gradually recovered over time, starting from day 8 all the way to day 30. We next focused on the function of the blood-brain barrier and visualized the cortex after injecting fluorescent dyes of different molecular sizes to assess vasculature permeability. We observed that the smaller 3K Dalton TRITC dye leaked much more rapidly across the blood-brain barrier compared to the larger 2 megadalton FITC dye. This finding suggests that the BBB was likely compromised following photothrombosis and microinfarction, resulting in vascular leakage. Interestingly, by day 5, the leakage of TRITC had returned to very baseline levels, indicating partial recovery of BBB integrity. So for our next investigation, we aimed to identify which cell types contributed to the recovery of vascular leakage around day 3 to the day 5 time point. Our results showed that the NG2 positive parasites here, you can see in the video, underwent elongation. And as I will show you in my next slide, it also went through division along the vascular cell walls following microinfarction. So these findings suggest that parasites play a very direct role in the restoration of the blood-brain barrier integrity along with recovery from strokes or infarction. So finally, you might be wondering whether our imaging platform can also be used to visualize not only disease progression and recovery in various models, but also the distribution and efficiency of different drugs. To address this, I've also prepared a few study examples in the next couple of slides. So here we injected a triple negative breast cancer cell line expressing GFP into a dorsal skin folder chamber implanted into an immunodeficient nude mice model. And on the left here, you can see the dorsal skin fold chamber implanted into dorsal skin or the back of the mouse. And the mouse has been secured using the chamber holder and mounted onto the intravital microscopy system. And after we prepared this and created the breast cancer model, 1 week later, we intravenously administered a nanoparticle drug coated with a substance mimicking selecting ligands to see its distribution, and then imaged the same area at 2 hours, 6 hours, and 24 hours post-injection. So over time, we found out that more drug particles moved closer to the breast cancer cells. And here we observed that the drugs, which were initially distributed mainly within or very adjacent to the blood cells, had reached the tumor tissue itself by around 24 hours post-injection. Although it's not shown in the slides here, we did also some additional experiments using drug conjugated formulations, demonstrating that these modified drugs exhibited significantly enhanced anti-tumor efficiency compared to the conventional forms of such drugs for targeting tumors. And so to conclude today's webinar, I'd like to highlight some main takeaways from today's presentation. So as mentioned earlier, intravital microscopy, or again, IVM, has transformed biomedical research by allowing real-time, high-resolution imaging of cellular activity in live animals. And intravital microscopy offers key insights into drug delivery, immune responses, and long-time disease progression. As technology evolves, intravital microscopy will remain essential for understanding complex biological phenomena and developing better treatments for human diseases, as well as being a very vital step in the development of drugs in the preclinical stage. And optimized in vivo imaging solutions for any organ or tissue, including transplantable imaging window chambers, accessory fixtures, and mounting parts, along with the holders, optimized for high quality observation. We also have developed a wide range of intravital imaging systems optimized for animals of various sizes, from small species such as mice and rats below 3 weeks, including larger ones such as rats above 3 weeks, and ferrets, guinea pigs, rabbits, and even non-human primates, using our robot arm system here, which is soon to be released into the market. And through IV Technologies' Intravital Microscopy system, which is optimized for in vivo imaging, we were able to capture imaging results from a wide range of organs. For example, shown today, the ear skin and the brain. But not only that, we can also see in the liver, the kidney, and again, the heart, liver, the heart, the lung, and even the lymph node and colon. So with that, I'd like to wrap up my presentation and we'll move on to the Q&A session. Thank you everybody for your attention.
Thank you, Jenny, for the great presentation. Now let's begin the Q&A session. So our first question is, how long the mouse survive with the chamber?
So it very depends on what kind of organ you have implanted the imaging chamber with. For example, the brain or the cranial imaging window chamber that I showed you today in my presentation, the mouse can survive up to about a year if inflammation is taken very well care of. And for another organ, for example, the liver, the mouse can live up to from 2 weeks to about a month, but this is also under the condition that inflammation has been taken care of very well in the recovery period after the surgical procedure for implanting the window into the organ.
Thank you. And the second question is, how do you label the blood vessels and cells?
So as mentioned in my presentation as well, we label the blood vessels and the cells by using in vivo labeling kits that are provided and developed by IVM Technology. And the base theory of these labeling kits is using antibodies that target certain biomarkers for blood vessels and for cells. And so if you have any interest in what kind of labeling kits we provide, please feel free to visit our homepage, or if you leave us an email at the email address you can see on the screen here, we will gladly send you our brochure and answer any questions you may have for the labeling kits.
Yes, and then the third question is the two-photon laser only used for second harmonic generation imaging?
So primarily, yes, it is used for imaging by utilizing the second harmonic generation, but it can be also very useful for imaging deeper areas in certain organs. For example, for the brain, yes, you can image brain areas such as the cortex using the confocal laser, but if you would like to image much deeper, then the two-photon laser would be very more helpful, as it has a longer wavelength, which allows it to penetrate into much deeper areas of the organ. So for deep brain imaging, the two-photon laser would also be very useful. And generally, if you would like to image deeper areas, not only in just the brain, but in other organs, using the 2-photon laser would be a very useful option as well.
Thank you. And it may be the last question. How are the in vivo labeling kits injected into the mice generally?
So generally, our in vivo labeling kits are injected intravenously. We mainly use the tail vein intravenous injection, but we also utilize the retroorbital injection as well. Unfortunately, we do have questions where they ask if it's possible to inject it through the muscle or through intraperitoneal injection, but it must be injected through the veins, so they are not used with intraperitoneal injection or muscular injection, mostly intravenous injection for the labeling kits.
Okay, thank you all for your questions. Maybe next step, the webinar recording will be soon available on our YouTube channel. For any further questions, feel free to contact us at information@ibimtech.com or reach out to our official distributor in India, Medi Analytica India. Before we conclude, we are currently conducting IVM demos at research institute and in university around the world. And we also provide customized virtual demos tailored to each researcher's interest. As I mentioned earlier, our IBM shop offers imaging accessory, forensic dyes, and training programs with special benefits for registered users. Thank you once again for joining us today, and we look forward to seeing you at our next webinar. Thank you so much.